ALS progresses more rapidly in some patients than others. New research findings have discovered that an overly active immune response, driven by a protein called interferon, appears to be a key factor in this faster progression. This response is carried out by specific types of immune cells, including those called microglia and astrocytes, which are found in higher numbers surrounding the motor neurons in the spinal cord. This connection between a hyperactive immune response and a more severe disease course suggests that this inflammation isn't just a side effect of ALS, it may be actively driving its progression. Instead of protecting these vital nerve cells, these specific inflammatory cells seem to be directly contributing to their damage. Microglia are a type of immune cell within the central nervous system which exist in distinct subgroups with different functions. In patients with ALS, researchers found two specific subgroups were more prevalent than in individuals without the disease. One subgroup, called MG2, showed evidence of impaired energy production, while another, MG3, appeared to be under significant stress, particularly in the spinal cord. These types of microglia appear to be actively involved in human patient ALS pathology. In contrast, a subgroup of microglia prominent in test mice with ALS called MG7, which is also an impaired energy producer, was notably lesser in the human tissue samples. The fact that MG7 is reduced in human tissue samples means that the mouse models, which are used to test potential therapies, may not be perfect representations of the human disease. A treatment designed to target the immune response by addressing the MG7 pathway in mice may not be effective in humans because their disease is being driven by MG2 and 3. This finding shows the complexity of translating laboratory research to clinical trials and explains why a promising therapy in a mouse model the same effect in a person may not have, but these findings provide a framework to developing future tests. While the study focused on microglia, their role as part of a broader immune response was also discussed. As the central nervous system's resident immune cells, microglia can contribute to the breakdown of the blood-brain barrier in ALS, which typically protects the brain and spinal cord. This allows other immune cells, such as T cells, to infiltrate from the bloodstream. Once inside, these cells interact with the activated microglia to create a destructive and widespread inflammatory environment. It's this coordinated effort among different types of immune cells that is believed to accelerate the degeneration of motor neurons. Understanding this full picture is crucial, as it shows that multiple interacting players are involved in the immune response in ALS, and future therapies may need to address this entire process, not just microglia alone. While targeting microglia is a promising approach, it's also a complex one. Research shows that these cells can play a dual role throughout the disease's progression. Specific microglia subgroups may be protective in the early stages but become harmful as the disease advances. This dynamic nature of the immune response means that therapeutic timing, deciding when to start a treatment and for how long, is a critical factor that researchers must carefully consider to ensure future therapy is both effective and safe. While this study offers valuable insights, the human tissue samples were taken from patients who had passed away, and as such provides insight of late-stage pathology. This is an important consideration, as research shows that the behavior of immune cells like microglia changes over time, so these end-stage findings may not fully represent what happens earlier in the disease. Furthermore, the study's discovery of key differences between human and mouse models highlights a significant challenge in ALS research. What works in a mouse may not have the same effect in people, underscoring the complexities of translating preclinical findings to effective human therapies.